All right, so let's kick off where we last started. Um, Looking at again monohybrid crosses, what that term mono is referring to is looking at just crossing of one trait, a mono trait. And in this sense, that's an example of what we were doing with the purple and the white flowers, looking to see what phenotypic trait, whether it be purple, whether it be flower, would end up being. Some other ones we might look at are seed color, seed shape, any of those other phenotypes we saw at the very beginning of this unit. A dihybrid cross, this is where I'm going to cross two particular traits. Not only am I going to look at crossing a trait on color, but maybe even on seed shape. So other than just one inheritance traits, I could also look at two. Now, when doing this, I can set up a different type of Punnett square, a Punnett square where I actually have up to 16 boxes. Now let's just draw one out real quick. Okay, when you draw out your box, this is not going to be overly pretty, but you can see that when we set it up, you can have up to 16 different boxes. Now, when setting that up, our phenotypes that we're going to be using should look still fairly significant, fairly normal. Now, if we selected a couple different phenotypes, let's just say we're going to use big P is purple, little p is white, and then I'm going to just choose a different color. I'm going to do big W. Let's not use that. Too much like white. Let's use big S for smooth and a little s wrinkled. And so what that's really referring to are my genotypes of two different traits. So I'm going to just give an example. If I have a seed that is heterozygous for his purple color as well as his seed shape, I'm already knowing that I'm going to be this, heterozygous for both. And I'm going to cross it with one that is going to be homozygous dominant for both. Big, big. Now what I need to do is I need to figure out what are my genotypic combinations that I can get from here. Now, this is where I talk about kind of like the foiling method, but let's do this side first. I want the best possible combinations I can get. I take the big P to the first big S. I take the first P with the second S, second P with the first, second with the second. 100% of the time, my only combination is big P, big S. However, if I do that same setup here, I got big P, big S, big P, little s, little p, big S, and little p, little s. Those are the different combinations I can get. So I take those four and I cross it with this one. And that's what's going back up here into the top. Okay, so if I zoom in on this, we have our crosses. We have big P, big S, big P, little s, little p, big S, and little, all crossed with this. So if I'm doing my Punnett square, there's my full-on Punnett square. I didn't have to do all 16 boxes, but I could have got my answer just from here. Now, if both traits, both individuals, going back to another one, if we had this, and both individuals are heterozygous for both traits, then we have to do they have to do a ratio. So if we had again big P, big S, little s, and cross with another big P, big S, little s, we have to go through and do all of our boxes. Just kind of zoom in a little bit to help us go through and fill it out. We're going to end up with a 9 to 3 to 3 to 1 ratio. And what that really refers to is that we're going to have 9 that are going to be dominant for both. 9331. 9 dominant for both. 3 dominant first, recessive second. What I mean by first and second is the first trait, so let's just say that this is going to be the P's and the recessive would be the little S's. This is going to be recessive for first, dominant for second. So this one would be little p, capital S's. And I'm only going to have one lonely old soul that's going to be recessive for both. Okay, that's our 9 to 3 to 3 to 1 ratio. That's what we're going to see when we cross organisms that are heterozygous for both traits. Okay, so let's take a look at what's going on here. So what's going on here? How are the alleles on different chromosomes handed out differently, together, or separate? So what this comes down to is, again, you're looking at our cross. We need to separate them out. We're heterozygous individuals. What do we end up getting? We want our allele combinations. And showing here which system explains the data. We want this one on the far right-hand side, which gives us our best possible combinations of our alleles. So if I just do a quick circle of that again, it's this one here we're going to look at together. So we zoom in on this. It's easier to see it. Remember, you've got your two crosses. These are both experiencing yellow and round seeds. Yellow and round. So it tells you yellow is the dominant, round is also a dominant. But in making the Punnett square, you can take a look and you see that the capital Y and R's, as they go all the way through, we get our allele combination on the top as well as on the sides. We should be able to do our 9, 3, 3, 1 count. Now, look at it again on the right hand side, 9, 3, 3, 1. I'm just going to cross them off based on 9 of them are going to be dominant for both. Dominant 2, 3, 4, 5, 6, 7, 8, 9. 9 dominant for both. Okay. I need to have 3 that are dominant for the yellow, but are wrinkled, so recessive. So there's 1, 2, 3. Now I need 3 that are going to be recessive for their greenness, and 3 of them that are going to be round and green. So I'm doing that. There's our 1, 2, 3, and there should be one lonely old soul again that is 1 wrinkled and green, and that's showing us right there. Again, only time you need to be looking at that is when we were heterozygous for both traits. So Mendel's laws of heredity. Number 2, law of independent assortment. Each pair of alleles separates and segregates into gametes independently, which means you don't necessarily know which one you're getting. 4 classes of gametes are produced in each one. So in this sense, that's why we have to do our full Punnett square to see which possible combinations we could have. You're not sure of what we end up getting. That's when we have to take a look at the qualitative data, look at the visual effects, the phenotypes of it, to see what we actually do end up getting. What meiotic event creates the law of independent assortment? It is meiosis 1. Okay, showing you here, we have our process. We go through the P generation, it separates out. In meiosis 1, principle of independent assortment, you start to see those organisms or chromosomes be pulled apart. Eventually, trace the genetic events through meiosis, you start to see the gametes fertilize and formation starts to come out. So a quick review of what we've talked about now. Meiosis 1 is huge because this is where we get diversity. Okay, law of segregation is, again, you've seen monohybrid crosses, but also in the law of independent assortment. Monohybrid crosses is segregation because it separates out individual alleles. But independent assortment is that there's multiple alleles that you could get. We don't have just one physical trait. We have multiple physical traits that we look at, and those traits can be independent from each other. Some we get from mom, some we get from dad. Why we get the uniqueness we do, why is the diversity of what we have today different from your siblings? That is because of the law of independent assortment, where 2 or more traits are going to be inherited down, and that is also going to be done in terms of meiosis 1. Okay, why do we use pea plants? Easy to grow, easy to see, quite a bit of quantitative data can be taken out of it. Mendel had strict control over what plants were being mated and which ones weren't, so he could make sure that there was nothing skewing results. It's another good reason why we had to use it. So good for genetic research. Relatively simple genetically in comparison to others. Hopefully that makes sense. If you guys have any other questions, please go ahead and jot those down on a separate sheet of paper. I'll should be back there be able to work with you guys on those when I get back. At this point, I'd like you to go ahead and go online. There are a couple different worksheets that you can be utilizing. Please go to the genetic problems set 1, where you can start working through monohybrid and dihybrid process. Thank you, and have a good rest of your guys' day.